Hello, everyone, and welcome to phyloseminar.org. This is the second talk in a series of 3 talks on the intersection of phylogenetics and public health. Please use the YouTube live comment box to ask questions. Today's speaker is Alli Black. Alli did her PhD in epidemiology at the University of Washington with Trevor Bedford, where she explored techniques for sequencing Zika viruses in the field and exploring viral genomic data to describe transmission dynamics. Graduating in April 2020, she used her knowledge of genomic epidemiology to help county public health departments in California use pathogen genomic data to support outbreak response to SARS-CoV-2. Alli is currently a senior epidemiologist leading the molecular epidemiology program at the Washington State Department of Health and the PI for the CDC-funded Northwest Pathogen Genomics Center for Excellence. In this role, she collaborates with academics and applied epidemiologists to translate new techniques in genomic epidemiology to public health practice and strengthen public health's ability to leverage pathogen genomic data. Welcome, Alli, and thank you for participating.
Thanks so much for having me. It's great to be here and so cool to have watched these philo seminars as a graduate student and now get to present one is a really cool experience. So very happy to be here. As Eric mentioned in my intro, I've been fortunate enough to work in genomic epidemiology research and now in a public health department building structures that enable us to deploy genomics as a tool for investigating outbreaks and maintaining situational awareness for surveillance. And so, I've been fortunate to really experience 2 ways really of doing genomic epi or 2 ways of using genomic epi. And so I want to share out what those experiences are like and explain applied genomic epi or genomic epi for public health practice, really from the standpoint of contrasting it with academic genomic epi or research genomic epi. And I hope that really kind of clarifies some of the similarities, but also the differences between more research-based versus public health-based genomics. So in terms of the structure of this talk, I'll start really by defining this terminology that I am using, academic versus applied genomic epidemiology. And I'll walk through some of the most significant or notable differences that I see between the fields as a way to really explain what I think applied genomic epi is and what makes it different. Then we'll talk a little bit of fun science, and I'll walk through an H5N1 outbreak that we had in the fall of 2024, highlighting not just the findings, but really what makes it different as an applied genomic epi study from what we might typically do from a research context. And then I want to finish off with some opportunities that I see for collaboration between academic genomic epidemiologists and research and the types of needs that we have in applied genomic epi, such that there's a lot of intersection there that I think would be really fruitful areas for working together. And this talk, I think, is for you if you are a student and you are wondering what a career in genomic epi in public health practice might look like. When I was a graduate student going through, this was still at a time when genomic epidemiology wasn't really fully operationalized in public health. And I've been lucky enough to get to work on creating those types of systems that do seek to operationalize genomic epi. And now we're at a point where we can share some of what that looks like. And maybe that'll interest you in terms of looking at careers outside of research. And for academics who are for sure in research, I hope that this provides you a sense of some of the needs that we have in public health and some of the data that we have access to, such that could inform potential research directions and ways that we can work together. All right, so moving into this terminology that I started off with at the top here, I like to talk about these 2 different angles of pathogen genomics in terms of academic genomic epidemiology versus applied genomic epidemiology. And I draw that terminology really from the background in traditional epidemiology, where this distinction is pretty common. So when you're studying epidemiology in a school of public health, we often talk about academic epi as like those beautifully designed, randomized controlled trials where you've spent years thinking about every possible bias and how you're going to design the study to control for it. Same thing for these super large cohort studies. Anything where you really have the time upfront to think about the best possible way to design the study, the best possible analytics for that study. And then you have this very constrained scoped project. And that really stands in contrast to applied epidemiology, where you typically have an outbreak. You need to figure out what is causing people to be sick. You need to do that quickly so that you can pull that potato salad off the shelf or what have you to stop people from being sick. And you tend to use less fancy methods. You tend to get things done a lot more rapidly, but then you tend to have to iterate and answer very similar questions over and over and over again. And I see the same types of sort of concepts in genomic epidemiology as well. So I think of academic genomic epidemiology as very much a research-focused endeavor. It is with a scoped, typically project-based analysis that has an end to it. And the goal often is to explore pathogen natural history or to uncover new knowledge about changing epidemiology or novel epidemiology or infectious disease dynamics. And that is a little bit different from applied genomic epidemiology, where we have this ongoing need to assess typically relationships between cases such that we can detect whether an outbreak is occurring, so that we can define which cases really are a part of that outbreak or not, so that we can provide situational awareness to policymakers and decision makers, and really so that we can inform interventions. And we are typically doing similar analyses or exploring similar questions very routinely because there's always another outbreak, in a similar context or with the same pathogen, what have you. So that's the broad strokes. And now I want to drill in a little bit more deeply into some of the key differences that really characterize this contrast. So the first one is really the types of data sets that we get to work with. So in academic genomic epi, typically you have data sets that are bounded by a data freeze. So often you are doing an analysis for a paper, and so you hit a point where you're like, okay, we're not going to incorporate new data past this time point. Here, I'm showing an example from Zika work that I did during PhD, right, where there's no data past 2018. It's not that there's no Zika anymore, but that is the constrained data set. And constraining it in that way then allows you the opportunity to do a lot of nice curation to get it into the perfect idealized form, where you don't have any really weird outliers off of the molecular clock when you remove any of those samples that maybe come from cultured isolates, if you have a direct sequence off of the specimen or any of those things like that. So highly curated, often manually so. And that stands in contrast to the data sets that we need to create and use in public health, because in public health, you have really this continuous integration, continuous deployment type framework where our avian influenza surveillance is never done, right? We're always looking for new data. We are continually rebuilding and redeploying our visualizations. We do that biweekly. And so, typically that's better handled programmatically. And so the types of constraints that you would put on the dataset curation are going to enable programmatic engagement with dataset curation. And you just have to be able to do it over and over and over at scale. And that's just a different need. The next big difference that I see are the types of questions that we explore in our genomic datasets. So in academic research studies, we are often talking about pathogen natural history or infectious disease dynamics. And I think one of the things that's interesting is that the tree might actually be like a nuisance parameter, sort of superfluous. So here I'm showing some figures from Gita Studis's analysis of the West African Ebola outbreak. This paper's from 2017. And, there is a tree there, but really the goal of that analysis is to look at the migration dynamics early on between Guinea, Liberia, and Sierra Leone at the start of the outbreak, understand where those introductions were going, how frequently they happened, when they happened. And so you're pulling that from the tree, but you don't really need the tree in and of itself to see that, right? Like the tree is a stepping stone. Same goes for skyline plots and looking at population size changes and factors associated with pathogen population size changes, right? Like the tree gets you the coalescent rates to give you the demographic, model that you have, but the tree is sort of, again, a stepping stone. And I think that is quite different from applied public health use of genomic data, where the tree is often the point. Like, typically we are looking at particular tips in the tree, particular clades in the tree. We want to know the relationships between those cases. We want to know whether cases we think are part of an outbreak are actually split between different clades. And so you see like these types of annotations of clade information or genotype onto other figures. So maybe you're looking at concurrently circulating different clades of a pathogen, or you're trying to understand the relationships between cases using genomic data. So in this figure here that I'm showing on the very right, like you're wanting to distinguish the fact that these cases of an infection that occurred in the office setting are distinct from each other and distinct from what we think the outbreak actually is, which is, whatever genotype is transmitting between folks on this assembly line. And so tree is usually the point and really focused on sort of relationships between tips. The next area that is pretty significantly different between applied and academic genomic epi is where we contribute to knowledge development. So I think public health gets a bad rap often for being antiquated or a little bit backwards. And absolutely, there are areas where that is true. I think our data infrastructure is really frustrating to work with. Like that's not an exaggeration. It is old. It does not necessarily work super well. There's a lot of feature development in terms of like APIs and things like that would be really nice to have that doesn't necessarily exist. The computational infrastructure provided for high performance computing, especially with Unix-based environments, is like a very, very hard one and hard to deploy. So like all of those things are true, but that doesn't mean that there aren't areas where in applied genomic epidemiology, we're really pushing the envelope or creating new knowledge. It's just that the areas where we do that look different than what we do in academic genomic epi. So, I think about academic genomic epi, we're often using advanced analytics, or we're potentially developing new phylogenetic methods or new evolutionary modeling methods to do interesting and new things with genomic data. We're often also detecting new or changing epidemiology. I think the canonical example of this that is really beautiful is using genomic data to realize that Ebola could be sexually transmitted from survivors months or sometimes even years after their prior infection, and seeing that actually happens with some frequency, and being able to do things like applying relaxed clock models to trees to look at branch specific rates and detect those types of events, right? I think that's a really exciting area of pushing knowledge forward of how pathogens behave. And then, there's a lot of integration of new data. I think you see evolutionary modeling integrated with transport data, flight data, mobile phone data, all of that sort of stuff to holistically understand what factors drive or correlate with pathogen transmission and infectious disease dynamics. And I think that is a very cool way to push the envelope forward. In contrast, the ways in which I see applied genomic epidemiology really creating new knowledge is more in this implementation science framework. So, what does it take to make these data actionable? How do you communicate the data in ways that provide decision makers with sufficient confidence that they feel enabled to take action off of the inferences you're making from genomic data? How do you, have methods that scale and work programmatically such that you can support this need to continuously integrate in new data and redeploy your visualizations and provide that analysis as this ongoing surveillance need that you just have to sustain? And then I think the last area, which I'm really excited about, I think it's still pretty new, but really is around genomic surveillance design. I think for both academic and applied contexts, so much of genomic epi has been founded on analyzing samples collected through convenience sampling. And, we've learned a ton from that. That's not to say that it doesn't work or that it's not useful, but, I think that there's certainly knowledge in the field that we could be sampling better, more proportionally, more representatively, and that's going to be necessary in order to get a full picture of the underlying pathogen diversity that circulates in a population. And that it will generally help our analytic methods if our data is adhering more to the assumptions of the models that we have. And I think we in public health have a really exciting remit and ability to partner with clinical laboratories or animal health laboratories, with outpatient clinics, to try and build the partnerships necessary to design systems for bringing sequence data or specimens to a public health lab to generate genomic data that are far more representative of the populations that we're trying to serve. And that really requires thinking about the factors as well that shape the pathogen population, that structure how we interact, how we spread disease, and ensuring that, within all of those substructured populations, we have visibility into that. So the figure that I'm showing here is just sort of a demonstration of this from SARS-CoV-2, where the figure up the top is the sort of proportion of sequences that we had from different counties in Washington, compared to proportional to the case counts that they reported. And after really working to partner to create new relationships with different labs throughout the state, the ways in which you can make representation of sequence data, or you can make sequence data collection far more representative of the state. So you can start to get data capture of what is circulating in rural Eastern Washington, which is typically a hard place to serve. And I think that doing this beyond COVID and really thinking about new paradigms for, say, respiratory disease surveillance is a really exciting area that we're just starting to explore in applied genomic epi.
If I may interrupt your flow a little bit, you're welcome to address this at the end, but my ears pricked up a little bit when you were talking about how do you convince the decision makers of the robustness and like the validity of your analysis, and or like that it's worth listening to, period.
I think a lot of that comes from showcasing how inferences from genomic data align or contrast with inferences that you make from epi data. So I think it is this mix of showing the consistency, like we thought that X happened given the case histories that we collected. We think that this outbreak is truly happening in this particular hospital because all of the cases, reported hospital stays at this hospital. And then having genomic data where, all of these samples are very closely related, the clade looks clonal or something like that, like having that congruence and being able to show it, I think like helps. Like they see like, oh, this is capturing something that we're also capturing with like our traditional surveillance mechanisms. But then also being able to show like where there are differences and talking about why, I think helps folks to understand what genomic data add. So, being able to I think differentiate samples that are very closely related versus some that are maybe like slightly less related, and being able to explain like what different hypotheses could be aligned with that particular dynamic in the tree. And I think really talking about it as like these are possibilities that are congruent with the genomic data. These are the ways that we could investigate it epidemiologically. These are the ways that we can investigate it genomically. I think really talking about it as hypotheses and questions to evaluate helps.
Yeah, it sounds like a complicated dance. You want to have it be similar, but like show that there's value add as well. That's cool. Okay, thanks.
Yeah, thanks, Eric. All right, so the last sort of point of distinction that I think is really important to point out, but is probably also the most clear to folks at baseline, is just like how findings are used. So I think when you're talking about academic studies, and this was certainly my experience coming through during PhD, the studies that we do are often like really cool, and we find really interesting things, and we basically just put it out into the world and hope that people run with it. And so, I'm showing here a phylogenetic tree of Zika virus. This is from the Faria et al. Paper in 2017, showcasing that Zika virus had circulated in Brazil and disseminated to other countries in the Americas like a year before Zika was ever detected. And so that is inherently useful. It tells you something about the time when people were at risk for a Zika virus infection, and therefore, if you're worried about monitoring for microcephaly or Guillain-Barré or any of the sort of potential outcomes of a Zika virus infection, you need to look at that time period as well. It tells you something about how well your surveillance system is picking up novel febrile illnesses that have a very similar symptom presentation to things that are more standardly circulating in your area, like dengue. But it doesn't actually like change any of the systems. Someone has to like read that and take it and run with it. And I think in contrast, in applied settings, the learning sometimes might be smaller, and the actions maybe aren't as like flashy or big, but they are often very, very direct. So here I'm showing phylogenetic analysis from a cluster of SARS-CoV-2 cases that happened in a congregate living setting in Humboldt County. This is in California. This is like summer of 2020. There was a lot of awareness that SARS-CoV-2 transmitted very effectively in older, vulnerable populations living in nursing homes and long-term care facilities. But it wasn't as widely known that you could get these large outbreaks in congregate living facilities with young people. And this was a housing facility for folks working on a commercial agricultural operation in Humboldt County. I think that probably sounds like weird to us now that we didn't know that or that we didn't realize that, but it was sort of new at the time. And so, the sort of response then is Humboldt County instituted a new testing policy for congregate living facilities. So they had a sort of more frequent testing protocol for monitoring for the introduction of SARS-CoV-2 into these settings. They did a lot more public health awareness and communications campaigns around the need for masking and hygiene, even for young people living in congregate settings. So small but very tangible impacts that change the way public health is practiced or how public health practitioners communicate about risk with the general population. And you have the people doing the analysis or making the findings like right alongside the folks who would take action on that. And that means that these things, even if smaller, can be much more tangible. All right. So those are the like big striking areas in my mind of how genomic epidemiology is different between research settings and applied settings. And so with that, I think if there's any questions, happy to take them. And if not, I'll move into talking about H5N1 in Eastern Washington.
That's great. Yeah, I mean, maybe you want to pause and say hi to your dog?
Yeah, I'm sorry. Can you
Totally fine. So people ask questions if you want.
Just close my door. All right. That should hopefully be better.
I guess I mean, I'm so much in the Linux and all the data is available to you world. I mean, can you just explain, I mean, and maybe you'll get to this, but can you just in a couple sentences describe like, what does your world look like? Or like, when you want to run an analysis, what does that look like? What is the infrastructure that's available to you?
Yeah, I mean, so I'll say like we are really lucky in Washington. I think, you know, we are definitely better supported and better resourced than many public health departments. When we want to run an analysis, we do have access to, you know, AWS, and we have, you know, our cloud-based Unix-based computing that we can run. We do also have like Windows subsystems for Linux on our local machines. I think so there is access to genomic data analysis. The part that's been harder, honestly, is we don't have the cloud access that we have is not sufficiently secured to move epidemiologic data or sensitive data onto. So we do a lot of genomic data analysis sort of in its own silo, and then through overlay, bring in epidemiologic data and anything that we need to really keep client-side or on our local machines. In a better world, we would have the scientific computing infrastructure sufficiently secured that we would be able to just do those analyses jointly. But like that, we're not there yet.
I'm sure it's a work in progress.
Yeah, definitely work in progress.
Okay, cool. Well, I feel like you should go on. That was a great answer to my question. Thanks.
Cool. Thanks, Eric. All right. All right, so back in October of 2024, we had an outbreak of highly pathogenic avian influenza, H5N1, on an egg-laying commercial poultry facility in eastern Washington in Benton Franklin County. And sort of as is typical with how this disease is handled, when you get detection on a poultry facility, and I'm stressing that because it's different from what happens with cattle, the poultry premise needs to be completely depopulated. They have to kill all of the chickens in order to prevent transmission and sort of wider dissemination of the disease. And what happens then is that typically, you know, independent workers are brought in to run the depopulation process of killing and the birds, and then also disposing of the carcasses in ways that are safe and prevent high path AI transmission outside of the premise. This is a really interesting dynamic that brings together different partners in these applied contexts. So, you know, for this outbreak in particular, on the animal health side, we have the Washington State Department of Ag, we have U.S. Department of Ag, and then we also have the Washington Animal Disease Diagnostic Lab at Washington State University that does sequencing for us on like so many animal specimens, which is really cool. But then on the human health side, when we have these large events and you have lots of people doing depopulation work, that's a really significant exposure risk. And so we monitor them to see, you know, hopefully to ensure that they don't develop H5N1 infections after doing this depopulation work. During this particular outbreak, though, as part of that monitoring event as depopulation was going on, we did actually detect cases in people who were doing depopulation. So we had 11 confirmed cases, 3 probable cases. All of those individuals had direct exposure to poultry. As part of this, when you get a case that tests positive, you also then monitor all of their contacts. You're wanting to ensure that if there is an instance of human-to-human transmission, that you're going to pick it up and hopefully, like, you know, control it really rapidly. We did not see any H5N1 detections in the contacts of the cases that tested positive. So from the epidemiologic data alone, the picture very much is that anyone who had an H5N1 infection had so because of direct contact with poultry, and that all of those cases are sort of distinct spillover events from poultry into the humans. The other sort of question is just like for those probable cases, those are individuals where their specimens tested positive at the Washington State Public Health Lab, but then their specimen tested negative at CDC. So they're not confirmed to be positive. And so that led us to wonder whether maybe some of these cases that were coming up as positive, but very weakly so, like could those be individuals whose conjunctiva or other swabbing spots were contaminated with virus potentially, but that they weren't necessarily infected. So already lots of different questions coming up. The other thing that you'll notice is there's lots of different partners involved. You have both the animal health side, you have the human health side, and then you have multiple agencies that you're trying to coordinate and communicate all between. That's both a very rich and fun experience and can also be convoluted and hard. And it's one of the things that really characterizes public health practice. So in terms of the genomic analysis, and specifically looking at genomic sequencing data of high-path AI viruses sampled from poultry on the affected premise, as well as from some of the human cases that cropped up, we wanted to understand whether this outbreak was a new incursion from wild animals or whether it was linked to a facility in a different state that was also experiencing an H5N1 outbreak. These were two distinct facilities, different states, but owned by the same parent company. And so we were wondering whether there was a connection there. So that was question number one. Question number two is this sort of weirdness around the human cases. Are they true infections, or are some of these possibly surface contaminated? Like you can imagine someone just, if there's a lot of virus in the environment and there's maybe a PPE breach or something like that, is it possible that they just have virus superficially on their conjunctiva or up in their nasal passage and they're not infected, but we're picking that up when we do our swabbing? And then lastly, big one for human health security, are the genomic data showing any evidence for human-to-human transmission, or are the genomic imprints consistent with what we're seeing from the epi data that we think that each of those cases are probably unique spillover events? So this is where we start. And I think the first thing is very fast and easy. We found that the outbreak strain for the outbreak that was happening in Eastern Washington was this newly detected genotype. It's newly detected at the time, D1.1. It was not B3.13, which is the strain of 2344B H5N1 that was sort of more associated with cattle transmission around this time. The other facility in the other state owned by the same parent company was having a B3.13 outbreak. So just the fact that those are different genotypes, I'm showing a tree here, but you don't even need the tree. It was okay, this is a distinct outbreak. And that's basically where you leave it. That is the point that is actionable. We didn't dig into this anymore. You probably wouldn't from a research context either. That is sort of just a nice cut and dry type thing. Where this starts to get a little bit interesting from a research perspective, but that we didn't explore from the applied perspective, is this question about where did D11 viruses come from? How did they arise? What are these dynamics? What was the reassortment patterns, etc.? So D1.1 was new at this time. It is a new introduction of 2344B H5N1 viruses to North America. Some of those introductions have come over the Atlantic flyway. I think other ones have come over the Bering Strait crossing. This looks like a new Bering Strait crossing. So if you look at D1.1 viruses, you see that they're sampled in 2022 in different regions of Japan. And then you can see just from the tips and sampling dates that There's sort of sampling of this genotype in Alaska and then into BC and into Washington, and very sort of characteristic of introduction and then migration through the Pacific Flyway. And I think, you know, if I had my research hat on or my academic genomic epi hat on, you know, I would really want to be constructing that history of movement, the timing of the movements. How long did D1.1 viruses circulate in different areas of the flyway before moving to a new area? Does that correlate timing-wise with migration, timing of different birds around these areas? What factors would have facilitated the Bering Strait crossing anyways? What did reassortment look like after entry to North America? Like, all of these different questions that I think would be really cool. I'd love to read a paper one day about it. But we didn't explore that because time and resources are constrained, and we are really trying to focus on the points that are actionable. So instead of exploring this natural history, we skip straight to looking at relationships between cases with a tree. And so here we're building an HA-NA concatenated segment tree. We can concatenate those segments because we are looking at D1.1 viruses, which we think don't have meaningful reassortment within them. And we are using HA and NA to try and maximize the amount of sequence length that we're looking at, but we're not using whole genomes because many samples don't have whole genomes. So this is very much like a data set shaped by pragmatism. And so up here, we see that we have human samples in red. We have chicken sequences in orange. And you'll see that our human sequences actually group into two different clades within the tree, which really surprised us. So all of these human cases did have exposure to the Eastern Washington outbreak. They did depopulation there. This sort of top clade, Franklin 02, is the main clade that we think really is associated with the Franklin County outbreak in Eastern Washington. So these two chicken samples here that my cursor is hovering over, those are pooled samples collected from the affected premise. Each sequence represents a swab of 11 chickens, so they're pools, and then you can see there's 4 different human cases that are grouping within that clade as well. Down here, this sort of Franklin 02 Oregon clade, you see only groups sequences sampled from humans that were collected during this outbreak event, and that they actually descend from chicken diversity observed in Oregon chickens. And that might give you a little bit of pause. We were wondering about this initially. Will often move around and these individuals had done depopulation at the Washington premise that we're talking about this outbreak in, but they were actually swabbed upon arrival, their first stay in Oregon. So there was a separate farm in Oregon that had H5N1 unrelated to our Eastern Washington outbreak. And these individuals had gone down to Oregon to do depopulation at that facility as well. On the day that they arrived in Oregon, though we don't know exactly when, Oregon sampled them, screened them, and found that they were positive for H5N1. And so, you know, from an epi perspective, they were defined to be part of our outbreak because, you know, that was the exposure that they had. And I think this is a really interesting, you know, coming back to Eric's question, this is a cool example of getting a little bit more information from the genomic data. So these individuals have competing exposures. They have an exposure in Washington. They also have an exposure in Oregon. And then the question is, like, where does this infection come from? Like, which exposure is relevant for the detection? And we would hypothesize here that, you know, the exposure that is more relevant to these specimens that were collected is probably the exposure in Oregon, because these samples are genomically more similar to strains that were circulating in Oregon poultry than they are to the strains that were collected from our Washington outbreak. So that's like actionable piece 1, is that you can define who is part of an outbreak associated with a particular exposure more sensitively with genomic data. How we can use that then also comes back to this question about contamination versus infection. So these individuals were sampled on the first day that they did work in Oregon, which is like not probably enough time for a true infection to occur and have enough viral titer to rise to the point of like having a positive PCR specimen. And so this is reasonable evidence, or you could imagine that this dynamic is maybe a little bit more consistent with possibly environmental contamination leading to some positives. Now, it's probably not true that all of these human cases are environmental contamination. Some of them had quite low CT values, quite high viral loads, and that would be more consistent with infection. In a perfect world, you would have serological information for all of these individuals, and you would look for evidence of seroconversion as your like gold standard as to whether they were truly infected or not. In reality, those samples are hard to get. You need an acute sample and then like another sample from someone a month out. These people are often lost to follow-up. So there's a lot of just like inherent challenges to public health and working with people and working on the ground. And so, there's no sort of slam dunks here, but this is enough evidence that it's possible that some of these cases could arise through environmental contamination, that we're now suggesting that when follow-up testing is occurring, if someone has an exposure while they're doing depopulation, that really that follow-up testing of workers, unexposed individuals, needs to happen before they start work for the day. So we are redesigning our monitoring systems to try and prevent any sort of confounding by environmental contamination. So that's like a very concrete action that we can take and have taken now in response to genomic findings. So the other thing that is sort of interesting and blends this work into the more academic genomic epi realm is then this challenge about sampling proportionally to disease burden. So, I think as is pretty typical for many studies, we focus a lot on sampling human cases and generating sequence data for human cases, and zoonotic reservoirs are often undersampled. In the case of H5N1, there's also a risk mitigation perspective to this. So from a diagnostic and response perspective, we know that if H5N1 is detected on a poultry premise, the entire premise will have to be depopulated. Like that is that. And so, there's no sort of diagnostic rationale for doing more sampling. And there's potentially negative impacts of doing that. So if you're doing, if you wanted to sample 500 birds or something, you have a vet in there sampling 500 birds and really increasing their exposure to H5N1, and you're trying to mitigate that. So right now, the sampling protocols are designed to, pool samples from chickens so that you have a higher probability of detecting H5N1 with minimal diagnostic burden if it's there. And then if it's there, you're trying to mitigate the risk to the person who's doing the sampling. So what that means is that poultry epizootics generally have very little sequence data representation, maybe one or two samples, usually consensus genomes derived from a pooled swab of 11 birds. And then you have sequences for like as many individual human cases as you possibly can if you have human cases. And so what I'm showing here are just, snippets of next strain trees. Whole genome is on top. This is an HANA tree on the bottom, which is why the numbers of tips differ. But what you can see right away is probably not going to be a surprise to some people on the call that the ancestral host state reconstruction on the internal nodes is, as you might expect, influenced by the numbers of tips sampled from these different games from humans or chickens. And so this is like right away a good case for using different analytic methods that are more robust to non-proportional sampling. So this moves us then to a structured coalescent approach, moving sort of a little bit more back into the academic world of fancier analytics. This is like not the most straightforward analysis though, because you can't use constant population size structured coalescent because you have an exponentially growing population. And then you still have these sampling artifacts that you have to overcome. So because you have very few chicken sequences, you're really not representing in the tree the poultry epizootic. Like you're not able to capture a different epidemiologic dynamic between wild birds, domestic birds, and humans. And so really this only works if you have a 2D model where you're just like, okay, what is in birds and what is in humans? Once we've figured all of that out, the reconstruction supports that each of these, this is a whole genome tree, which is why we only have 3 human samples in here, but that each of those human cases is a distinct poultry to human spillover event. And we've also looked at the minor allelic variation in the pool and done some like direct data analysis that I'm not showing here that is also consistent with that. This is also consistent with the epi data. So we're pulling together multiple streams to think about this. But to me, like this is the academic genomic epidemiological solution to this problem, right? Is like use as fancy analytics as possible to try and overcome a really kind of hard data set and do your best. And the applied epidemiological response in my mind really is we need to rethink sampling design and design sampling frames or sample collection to align with genomic endpoints. And so like, that's thinking about improving representation of what is circulating in wild reservoirs. That is capturing poultry epizootic diversity dynamics with more sampling. And really contextualizing human sequence data within the variation that actually arises in a poultry epizootic. And so, rather than just handling the situation that you have with fancier analytics, like can we actually design systems that collect data in a way that are closer to the analytic assumptions? And I think that latter path is really important for health departments that are maybe coming into genomic epidemiology more recently, who maybe don't have folks with expertise about Bayesian phylogenetics and structured coalescent analyses and things like that. But if we can collect data in a way that is more amenable to analysis with the rapid tools, with the easier tools, like I think in the end, that's going to lower the barrier of entry to this type of work, and that'll be important. So just to kind of wrap this up in terms of like what is the actionable work that comes out of an investigation like this, I think one of the first things is really just like unifying communications to both human and animal partners. So in this type of scenario, you have an incident management team that is stood up on the animal health side. And that is completely separate from the incident management team that is set up under the emergency response framework to respond to like the human cases. And those don't really have points of crosstalk unless you make them. And so I think one of the really cool things that we were able to do is bring together through partnership the genomics team that works on the animal side at the Washington Animal Disease Diagnostic Lab, as well as the genomics team at Washington Department of Health, and really think about the data together, integrate our own perspectives and expertise from veterinary health and human health, and come up with joint communications plans that then could be disseminated out to both sides of the emergency management systems. And so that's a very different thing that arises. And then you have communications and talking point and situational awareness that's shared in real time. I think we also mentioned this, that there's some at least preliminary evidence that some of those positive human cases might be the result of environmental contamination. rather than true infections. We can't answer this perfectly. We don't think that all of the human cases are from contamination. We think some of those probably are true infections. But going forward, we've changed our guidance around when sampling should occur for that monitoring process, and that should hopefully mitigate some of this issue moving forward. So that's like a direct change that we've made to how we'll respond to these types of events in the future. The other thing that is very clear is that like the wild epizootic is undersampled. One of the things that we can do and that we're starting to do is explore what it would look like to improve surveillance coverage. Again, this is a lot about relationships. This is reaching out to like Department of Fish and Wildlife to understand what types of surveillance activities they do. What would it mean to sequence them? Like where would they need to go? What does funding look like, right? Like there's a lot of pragmatism, but there's a very concrete thing that we could do, funding permitting and partnership permitting, to change the access to data that we have. And I think along those same lines, when it comes to understanding the poultry epizootic and the dynamics that are happening there that we're completely missing, we could explore what it would look like to make changes to the sampling protocols that are in place that are designed for diagnostic endpoints, but not necessarily genomics endpoints. Whether those get adopted or not, I think is sort of, a standing question, but we can at least propose what it might look like to have a different sampling protocol for these scenarios, such that genomic data sets going forward when we're investigating H5N1 outbreaks are more in line proportionally to the actual disease burdens that we're seeing. All right. So that's kind of everything I have on H5N1. Happy to take any questions about that before wrapping things up with like points of collaboration.
I have questions for the end, but
Okay.
Yeah, I think you're good. Thank you.
Thanks. All right. So just ending with things I would like to see happen to bring applied epi and academic, or applied and academic genomic epi a little closer together. So I think the first one that would be amazing to have would be an improved understanding of how sampling frame impacts analytic methods. This is a figure from Matthew Hall's paper exploring how different sampling schemes affected SkyGrid population size reconstructions in Bayesian phylogenetics. I would love to see this kind of thing for, say, phylogeographic inference, right? So this, the H5N1 is a good example of DTA really not working because the assumptions about numbers of tips in the tree being representative of disease burden is so violated. But there's a spectrum there, right? And I think it would be incredibly helpful to have more work and more simulation-based analysis that explores how robust these different tools are to different sampling frames and like when they break down. I think the other thing is that like this paper showcased that proportional sampling and uniform sampling perform similarly well. And that's really helpful knowledge to have as an applied practitioner, because then when we're designing these systems, we don't have to try as hard to get like perfectly proportional sampling. We can focus more on getting uniform sampling, which is an easier ask, practically speaking. And so just having that interplay between like, how do we design these systems? What sorts of sampling frames are important to design for? And then like, how will that affect our inferences is an area we really should be exploring together. And then the other one I think is really this question of unifying measures of disease frequency. So if you're doing Bayesian phylogenetics and looking at population size changes, those estimates are given out in any tau. The figure here that I'm showing is a similar thing, maximum likelihood framework from the Kleist lab in Germany, and estimates pathogen population size, this phi-est parameter, and can correlate it with reported cases. But again, they're different. And that is, I think talking about relative increases and decreases and timing of relative increases and decreases in pathogen population size is a helpful first step. But I think it will be far more useful and far more easily applied and understood by traditional epidemiologists if we can actually, take something like any tau and translate it more easily into incident cases or prevalence. I think there's kind of multiple challenges here. I think there's a little bit of discussion in the literature as to whether any tau is really giving you sort of prevalence versus number of incident or new infections. And then there's also the question of just like, how do you do that translation in an ongoing way that you can do kind of all the time? It's robust. But I think that this would be an area that would just really help with adoption of phylogenetic tools. And I think would fill a huge gap because case surveillance data is really affected by underreporting or asymptomatic infections. And if we could get more information about what is actually happening from patterns of genomic diversity, and we could explain that in the terms that traditional epidemiologists use, I think it would be like profoundly impactful to how we do surveillance. So this is also an area that I really hope is strengthened. There are others. This is not exhaustive, but those are the two that are my big ones. So if anyone is interested in working on these questions, please don't hesitate to reach out to me. I'd love to hear from you. So that does take me to the end of everything I had prepared. Thank you so much for having me. That is my email if you'd like to reach out to me. And yeah, happy to use the last few minutes for questions.
Great. Well, I think that was an awesome talk. And I was actually going to ask specifically about the last section of your talk. So you gave it. Before, so please post questions, people out there. Can you just clarify, you used the term sampling frames several times. I thought maybe you could just, this is a term of art, so maybe you could just explain it briefly.
Sure. I think of sampling frame as how you are trying to sample. Like, are you, what factors are going into your sampling design? So I guess that would be the best way to that I would say that. I mean, I think convenience sampling is a sampling frame, right? It's just whatever we can find. Proportional sampling is a sampling frame. We're gonna, and you would define it a little bit further in terms of proportional to disease burden, proportional to population size, but really defining how are we doing sampling, I think is the key there.
So with respect to your second sort of to-do for the community, the connecting the, So can you just be a little bit more concrete? What is it that you want? I mean, so there are people that are working on methods that take both sampling data, information about case counts and genomic data. And so you would want ways of contrasting these, or ways of doing inference that uses both these sources of data, or what would be your dream thing to have?
In my absolute dream, okay, so in my absolute dream, we probably don't have the red line, which is reported cases. In my perfect dream world, phi-est, which is taken from the genomic data, actually gives me these counts as reported cases, rather than as some sort of weird amalgam. And I think that, that there's this question of effective number of infections versus true number of infections. But then there's also a question of do you need to vary serial interval tau over time or not?
Let's start with that first one. So you're saying you feel happier with the phi estimate, and you want to be able to pull the red line towards the blue line.
No, sorry. Okay, yeah, I'm not being clear. I would want phi estimate to actually be number of cases. I would like to, because like right now, if I didn't have reported cases, which would often be true, or it wouldn't be scaled correctly, I would talk about sort of, oh, well, I see that phi-est is peaking around January 2021, and then it falls off. But then like the state epidemiologist would be like, so what is that? Like, how many infections do we have? And I would be like, well, I don't know. I know that it was, and if I had multiple years of data, right? Like, okay, if we looked at Scotland, I could say like, well, I think, basically right now, the number of cases that we have looks fairly similar to the number of cases that we had in April 2020. And I could talk about this in terms of like relative comparison in terms of how much disease burden is out there. But what I'd really like to do instead is be like, we're having a surge. We're having like 100 new cases detected every day at this point, given the patterns that we're seeing in the genomic diversity.
Okay, and so you want to be able to use, if I understand correctly, you want to be able to use some case count data to calibrate what the phylogenetic techniques are telling you in terms of like actual case count.
I think that building these models, like these calibration, like you need this data in order to assess that the genomic data is providing a reasonable assessment of case counts. But I think in the ideal world, case counts are often incomplete, right? Like you either have subclinical infections that you don't see, you have asymptomatic infections that you don't see, you have simply like infections that are occurring that you don't see because people don't have access to care. And just none of that gets reported. But, presumably the history of those infections is still captured evolutionarily. And so the genomic data has the potential to tell you that story in the absence of the surveillance data doing that. And I think if you could harness that, it would be really helpful.
Okay, cool. Well, you were continuing before I interrupted you. I can't remember where you were going. Is that?
I think that was it.
Okay. So yeah, that, and also handling like weird sampling and just like vagaries of real data that you get when you don't get to pre-specify what it is that you want.
Yeah, and like really understanding like how robust are these methods? Like I think if you do sensitivity analyses on discrete trait reconstructions, like often even in the absence of beautifully designed proportionally sampling, it works just fine. But actually knowing like how much sampling bias can it take before it breaks down, like having that sort of simulation-based assessment would be really useful.
Yeah, that's interesting. And you feel happy enough with the current simulation frameworks that like having simulations using those types of frameworks would convince you?
I guess maybe I'm not thinking about it fancily enough. I'm thinking you could do stuff with just like simulate, like you could just take an outbreak that you simulate and sample different levels of proportionality and just infer trees and look and see at what point it breaks down. I think that would work. Yeah.
That's awesome. So it's interesting. So those are both like, I feel like on the gradient of like all the different types of methods, those are sort of more a little bit on the fancy exotic side of things. I thought maybe you were just going to be like, tell everyone like, all y'all like stop all this fancy stuff. Like what we need is like, it's like you're proposing like a nice luxury automobile, or I don't know. It's like out there. I thought you were going to say like, look, all we need is a cart or something.
Oh, well, I think we need, I think often, what public health needs to do its job is maybe a cart, right? Like, I think, this gets back to the, like, in the end, we do a lot of public health work simply by looking at where tips fall in the tree. And there is a lot of work that is done there. But I think for our work to have more utility and to grow the impact of using genomic data in public health practice, like there is the need to have a more robust understanding of like how these analyses perform, or there is new capacity that is needed. And likely that type of research and development work is not going to be done in a public health context. So my hope is really that academics are interested in these questions and can kind of push things forward, and then we can have more of this translation piece back into public health. But I think like we wouldn't necessarily be the people doing the simulation-based inference to like do these assessments. I mean, you like theoretically we could, but there's just like not the time and bandwidth.
I mean, you're handling stuff on the ground.
Yeah, exactly. Yeah.
The other thing that sort of sticks out to me is like, I mean, the thing that's amazing is that you have control over your sampling techniques. And is there a role for suggesting optimal sampling strategies, or is it just sort of so obvious that you would just, you just think about it and you just do it?
No, this is totally one of the gaps that we have right now, and I just didn't put it in here for the sake of time. But if you look at traditional epidemiology, there is a really strong framework for like powering analyses, how many samples, like to answer different questions, and like none of that exists for genomic epi. And like if you ask anyone like how many samples do you need, right? Like someone's going to say like enough. And I think Shirley Wohl has done work on this, but I think we need a way better either theoretical framework, or just like if it is a simulation based thing where like you have to simulate what things are going to look like in order to know how many samples you need, then we would need a sort of simulator that we could run easily to do that work. But I absolutely think that in terms of like which samples, which structuring variables, how many samples to collect over what time frame, like all of that, like if we could more rigorously test that and then provide guidance as to how to do that design, that would be really helpful.
I'll reveal my biases, which is like, I mean, analytical power analyses are like very hard and they have a lot of assumptions. So it's interesting. I mean, I feel like there's some real low-hanging fruit here. Like we need to have some good simulation frameworks that are very transparent about what their assumptions are, and then have some automated pipelines for doing the analysis and following up and figuring out what's going on. I hope somebody picks that up. I don't think it's quite in our wheelhouse, but I feel like I'm almost tempted.
Somebody please pick it up. I think that would be amazing. And then I think, pretty much every public health department, I think, used like Shirley Wohl's framework for SARS-CoV-2 specimen numbers needed to estimate prevalence, those sort of calculators. So like, yeah, if you want a thing that is probably picked up by like every public health department in the US, like build a way to like, design how many samples you need to answer particular questions or do particular work.
That's super cool. Yeah. All right. Well, everyone go. Well, thank you so much, Alli. I really, that this is exactly what I hoped you would talk about. So I really appreciate it.
Yeah. Thanks so much for having me. It was really fun.
All right.
All right.